Hello, everyone. My name is Amanda Miklas, and thank you for joining our webinar today entitled Manufacturing Challenges Facing Cell and Gene Therapy, which is being co-hosted by Informa Pharma Intelligence and Alliance for Regenerative Medicine. Gene therapies and cell therapies are now viable medicines that have reached the market, providing transformative benefits in terms of patient responses and meeting large unmet needs. Manufacturing is at the forefront in this sector as the manufacturing process is the product. Even so, we're still in the early stages of this current generation of regenerative medicines, and because development has been accelerating at such a rapid pace, there are many manufacturing issues at hand, such as production capacity, logistical challenges, and standardization that still need to be solved. Today, we're looking forward to hearing directly from preeminent sector experts on their detailed insights and industry best practices regarding manufacturing and industrialization hurdles unique to cell and gene therapy. Before we begin, there are a couple of housekeeping items that I wanted to mention. If you would like to submit a question during the webinar, I direct you to the box below the Media Hub on your screen where you can ask a question, and we will hopefully leave some time at the end to get to some of those questions. In addition, if you are having any technical issues with the webinar, please use the same box as well to contact us. This webinar is being recorded, and that audio recording, along with a copy of the slides, will be sent out to all of the registrants following the webinar. So to begin, we're first going to go through a round of introductions to our panel. Again, my name is Amanda Miklas. I'm an analyst and senior consultant with Informa Pharma Intelligence. And for the past several years, I've been focused a lot on tracking events and trends in the regenerative medicine market, particularly pipeline trends, pricing and reimbursement issues, as well as manufacturing. And I'm really excited to be moderating this webinar today. So we'll just go around the virtual room here, and as your picture comes up, if each of the panelists would please introduce yourself and provide a little bit of your background as well as your current roles and responsibilities. So we'll begin with Mike.
Thank you, Amanda. My name is Michael Lemke. I am the Director of Science and Industry Affairs for the Alliance for Regenerative Medicine. Prior to being at ARM, I was with Johnson & Johnson for 20 years in research and development, focusing on tissue engineering, biomaterials, and human cell and tissue products. My role at ARM as the Director for Science and Industry Affairs is to work through our technical committees with our members on key issues they're facing, among which are our challenges in the area of manufacturing and CMC, some of which we'll talk about today.
Thanks, Mike. Next, David.
Hi, this is Dave DeGusto. I am Chief Technical Officer at Sema Therapeutics, where I'm responsible for CMC activities around manufacturing, including process development, manufacturing, quality systems, and regulatory affairs. Prior to joining Sema last August, I have spent 25 years in academia and 9 years in industry developing cell and gene therapy products, including translational research, process and product development, and execution of clinical trials.
Thanks, Dave. Next up is Michelle.
Hello, thank you to those who have joined and ARM as well. My name is Michelle Berg, and I rejoined Aldevron recently to serve as the president of the GMP Nucleic Acids Business Unit. During my 4-year time away from Aldevron, I had served as the VP of Patient Affairs and Community Engagement with Abeona Therapeutics, a gene therapy company. Aldevron is a CDMO serving the biotech industry with our custom production of nucleic acids, proteins, and antibodies. Thank you.
Thank you, Michelle. And last but not least, Bob.
Hi, everybody. My name is Bob Preddy. I'm the Chief Executive Officer and President of Hitachi Chemical Advanced Therapeutic Solutions, which used to be PCT. PCT is now the global brand for Hitachi Chemical's regenerative medicine business sector, which is now, we're set up now in Munich after having purchased Abseth in April of this year, and we've got a plant in Yokohama. We're currently at about 180,000 square feet of manufacturing and development space around the world, about 500 people. And I've been doing this since I co-founded the company in 1999. Prior to that, I had spent 13 years in various roles, one at the New York Blood Center in this industry directly related to bone marrow transplant, where we worked with purging and processing engineering of grafts for bone marrow transplant field. And prior to that, I was with one of the first cell therapy companies before we had the name cell therapy, Marrowtech, which became Advanced Tissue Sciences and produced Dermagraft from 1986 through 1989.
Thanks very much, Bob, and thank you everyone for those introductions. Next, I am going to turn it over to Mike Lemke from Alliance for Regenerative Medicine, who's going to provide us with a brief update on the regenerative medicine sector, including why addressing manufacturing challenges now is such an urgent issue. So I'll hand it over to you, Mike.
Thank you, Amanda, and thank you everyone for joining us today. And special thanks to my fellow panelists. We have a real wealth of experience on the panel today, and I'm looking forward to a fascinating discussion. But first, what I'd like to do is set the table for you on and describe why we at the Alliance for Regenerative Medicine think that manufacturing in this sector is at such a critical stage right now. So I'll start on the first slide with a few quotes from the heads of our regulatory agencies. This is former FDA Commissioner Scott Gottlieb and current CBER Director Peter Marks, and they've been quoted as saying that they expect that there will be more than 200 INDs per year in this space by 2020, and that will translate into 10 to 20 cell and gene therapy products a year being approved by the FDA by 2025. Guido Rossi, who's the director of EMA, made similar comments during his remarks at ARM's 2019 meeting on the Mediterranean, to the effect of expecting to see 10 to 20 product approvals or submissions each year within the next 5 years. Some of the numbers that are driving this, we're tracking currently over 1,000 clinical trials at ARM, along with our partners at Informa. These are phase 1 through phase 3 trials, the majority of which are still at phase 1 and phase 2, but a number that's not an insignificant number have moved into phase 3, and we expect many of those to translate into commercially successful products in the coming years. These are in the area of gene therapy, gene-modified cell therapy, which we define as ex vivo modification, genetic modification of cells, cell therapies, and tissue engineering. Moving to the next slide, one trend that we are seeing are indications with larger patient populations moving into clinical trials. These are in areas such as cardiovascular, central nervous system, and diabetes. The first gene therapy for critical limb ischemia was approved in Japan this year, and there are trials ongoing in diabetes, Parkinson's disease, stroke, and other indications that have significantly larger patient populations than some of the products that have been currently approved. Another factor at play is the importance in this field of expedited approval pathways. So there's currently 34 therapies that have been given the RMAT designation, 18 given breakthrough designation, 34 given fast track, and in Japan, 4 given the Sakigake designation, and 19 prime designations in the EU. These are primarily in the area of gene and cell therapy. The important thing here, and why this is important, is that we can see from 2 current examples, Kymriah and Yescarta, that they were able to obtain approval by an accelerated pathway. and about 2.5 years from the initiation of their pivotal study to approval. The comparable number for a small molecule is about 9 years. And traditionally, a lot of the heavy lifting in the manufacturing and CMC area has been done in parallel with those phase 3 or pivotal studies. So what we're seeing is that there's a benefit here, obviously, to these accelerated pathways in making these products available to patients earlier. But it also presents a challenge for manufacturing, as there's much less time to deal with issues related to scale-up. Here's another look at different indication areas that currently have products that have obtained expedited approval. You can see the majority of those are in oncology, but also a significant number in other areas where there are larger patient populations. So with that, I will conclude my initial comments and we will begin with the panel discussion. So I'll turn that back to Amanda to start that.
Thank you, Mike, for sharing that information and data. It was really informative and interesting and exciting to see where the sector is moving in terms of growth. So thanks very much for that. So as Mike said, next we're going to move on to the discussion points for this webinar. We have several different topics that we'll go through one by one, hopefully getting to all of them. And first topic for our discussion today deals with transitioning manufacturing from smaller to larger scale. So manufacturing methods for gene therapies have traditionally been established at academic centers where production has been at a much smaller scale, and this is sustainable if you're producing smaller batches for early stage clinical trials. But as the gene therapy and cell therapy pipelines have been advancing as a whole and quite rapidly, particularly because of the expedited development pathways that Mike has just highlighted, sponsors need to be able to scale up quite quickly to support larger scale manufacturing as those therapies move into mid to later stage clinical trials and eventually and hopefully commercialization of a viable product. So I'd like to get the entire panel's input on what you think are some of the best practices for sponsors to move from a smaller or pilot process to a larger or commercial process. What are the biggest challenges in doing that, and what are the best strategies and some of the lessons learned in your experience? So I'll turn it over to the panel and for anyone who would like to begin with that particular topic.
Well, this is Bob. I'll start. Michael set this up really well by talking about the geographic expansion. The fact that each company is developing therapeutics, more and more therapeutics, and there also this geographic expansion, breaching boundaries that now go across regulatory frameworks, various regulatory frameworks. And at the same time, there's these accelerated pathways that are that most of these therapies will be coming out of phase 2 now and going into commercial production. So the speed at which the CMC needs to catch up to the clinical data is really inconsistent in many ways with where we are as an industry, and this presents one of the biggest challenges. So creating that balance that's needed to develop manufacturing in a phase-appropriate manner, taking into account accountability risks, key commercial drivers, the investments required, et cetera. We see imbalances in both directions from a contract manufacturing standpoint. We can see many of these products. And people are looking to automate. You can't really automate too soon. Can't wait too long. So really getting that balance and automate or innovate is probably a better way to look at it. Getting that balance is really going to be very important, and understanding where the risks are. And I'll just close this piece, but there's other time to talk about this, but we see the risks in 4 big categories, the lens that we view all of these processes, with quality, robustness being one part of the lens, cost, scalability and sustainability, and needing to look at each one of those for each part of the manufacturing process. So at the end of it, it really just takes a plan and not heading headlong into this saying, I hope we get approval, because approval is one thing, but actually distributing these products in a sustainable way is what the goal is to get them to patients.
Great.
Hi,
Thanks, Bob, for your thoughts. Oh, go ahead.
This is Dave D'Augusto, so I'll make a couple brief comments. In terms of transitioning from smaller pilot scale production to larger commercial scale manufacturing, the challenges can vary depending on whether there are autologous or allogeneic products involved. So in one case, it's large numbers of iterations of small culture systems to make patient-specific products, and in another case, for example, regenerative medicine, it's going from small pilot scale to making large banks that can be used to produce products from a single source, and they have different challenges. I think those challenges have different aspects. For example, in many cases, small bioreactors to make patient-specific products come from a single vendor, and so if you want to go from a 20 to 50 patient pilot study or registration study under RMAT and move into making products for hundreds to thousands of patients, one of the real questions is, can the equipment providers keep up, both in terms of manufacturing the equipment as well as providing the disposable sets that are used in those equipment. So I think there's a lot of issues there in terms of the equipment supply. But the same thing goes for scalability of the supply chain. So again, if you're doing small trials, the amount of media you need, the amount of growth factors, cytokines, small molecules may be very modest, and there are certainly a number of small companies that have provided niche services in this space. But as you want to grow to a commercial scale production, you may or may not be able to get those companies to scale accordingly and maintain the quality of the reagents you need. And then finally, as I'm learning now, as you scale up, the composition of matter and the reproducibility of productivity at scale is not simply linear. There's a lot of issues related to growing things in larger bioreactors or at larger scale that you can't just simply multiply by volume and assume you'll get the same yield. So I think those are some of the issues that have come up that have kind of keep some of us awake at night when we're thinking about getting to commercial stages.
David, with SEMIS technology in particular, I understand it involves encapsulating stem cell-derived islets in a device. Are there special or unique scale-up considerations in terms of that, in terms of?
I'm sorry, I lost you at the end there.
So SEMIS technology involves encapsulating stem cell-derived islets in a device. So are there unique scale-up considerations for that in terms of sourcing and or the quality of the materials?
Yeah, so there are two different aspects. So on the cell side, since we're deriving cells from embryonic stem cell banks, we can do scale-up, which is formally larger production campaigns from bioreactors. But on the device side, since those are patient-specific, not patient-specific, but each device is implanted into a patient, it's a scale out of number of devices, and certainly going from our prototype device models where we manufacture these in-house for early clinical trials, we will be going to sort of mass producing those, and so there's some challenges with identifying both the methodology to mass produce as well as the capability to mass produce. So I think one is iteration of number of units of devices. The other one is scale up of actual batch manufacturing for the cells that fill those devices.
Mm-hmm. Okay. Thank you. Anybody else on the panel want to comment on this particular topic?
Hi, Amanda. Excuse me, this is Michelle speaking. So something I just wanted to add, this is an important topic with regard to the considerations that are taking place early on, even before a sponsor gets involved necessarily. As it was shared, at this point, by and large, much of the innovation is taking place at the academic centers. And again, their goal is not necessarily the view to commercial. It's really just to get to that phase 1 study. And so some of the choices and things that are being made at that early stage really tend to lock people in. And so I think if we are focusing on those centers and helping to educate what tweaks and changes they might be able to make early on to enable that path to commercial, that will help the field advance much more swiftly, and in this environment where CMC has such a high focus and is moving very, very quickly. And the last thing I'll comment on is it's such an important topic that we'll be talking about this even further at the upcoming Alliance for Regenerative Medicine meeting on the Mesa as one of the morning workshops.
Okay, great. Thanks, Michelle. All right, then we'll forge ahead to the next topic here, which concerns a debate that I've heard a lot about at different conferences I've attended and read about in the literature, which is from a therapeutic developer's point of view, making the decision or making the call on whether or not to build out manufacturing in-house or to outsource that, or partner with a CMO or CDMO. And the main takeaway that I get from this debate is that there's no right or wrong answer. It's not a black and white answer, and it's really highly dependent on the individual company that's involved and where they are in their particular life cycle. But given that, I'd love to hear the entire panel's insights on what you think are the advantages and disadvantages to building out manufacturing internally, building out a manufacturing facility or leasing out a manufacturing facility, as opposed to outsourcing and teaming up with a CMO or CDMO. So pros and cons to in-house manufacturing, as well as pros and cons to outsourcing manufacturing. So I'll open it up to the entire panel for comments on that.
Again, I feel like, at least this is Bob. I mean, at least with this question, since I'm connected with a CDMO, it's probably I'll start the ball rolling on this. We recognize that the audience will take statements by the leader of the CDMO enterprise with a grain of salt here. So I think I'd like to reflect on what we see mostly on the second part of this question, basically, what is the right time to consider the choice. And I think you're right. There is no right answer here. We have companies that look to insource the discovery engine portion, big companies and small companies alike. We have companies that look to outsource the commercial part. And I think it'll end up being a blended approach that will depend upon a few factors, not the least of which is, where outsourcing providers offer platforms that offer additional value to customers. There's a potential to make outsourcing a win at even high volumes. Where that's not the case, there's a mathematical decision to see which approach is best, say the most cost-effective. We see speed to market as a consideration, as CDMOs have the capacity to set up, and often people would like to get going quickly, and we've seen good examples of that in the case of Kymriah particularly. Geographic considerations will weigh heavily in this decision. A company may wish to put its own manufacturing footprint in one portion of the world, but look to outsource in other portions of the world. So it doesn't need to be, doesn't have to have the responsibility of building and maintaining and trying to keep these manufacturing facilities full all over the world. And so in the end, a blended model, different customers, different products, different regions, how many products, how complex the products are, what's the volume of distribution. It's really too broad a question to say at which stage, but it's really more, how can each stage be approached by the right business model. And all of this is in development right now. There just is no right answer, no one size fits all.
Mm-hmm. Okay. Yeah. Right. Bob, one of the key buzzwords I keep hearing in discussions, in this particular debate is cost of goods or improvements in economies of scale. Is that a key advantage from a developer's point of view to outsource and work with a CMO or CDMO?
Well, I mean, that's one of the big questions. First of all, I mean, I have a slightly different view to what you hear a lot, which is that the cost of manufacturing is driving the pricing. I don't think it is. I think the reimbursement of these products is really being driven as a value-based reimbursement mechanism. Right? What are these therapies worth, particularly if they're one-time cures? And that doesn't mean we want to reduce the manufacturing cost of goods, and we do. But looking at it from the ground up, which is labor, materials, supplies, overhead, those sort of things. If we just build that up for the X amount of hours it takes to manufacture and test a product before it is shipped out, there's a cost associated with that, which is fairly equal between a CDMO and in-house manufacturing. That doesn't really change. Where the cost drivers come in are how well you fill the capacity that you build. And that's a question, again, of how long are these products in the incubator, what time are they taking, how many people, what are the drivers, and what's the volume? And the difficult thing to do is it's like catching a knife, right? When you first put a therapy on the market, there's typically a backlog of patients that will receive them, so you need a lot of product. Then you'll hit some peak, and then it'll start to come down again once you get those the backlog of patients gone. And for the one-time cures, you'll start to see a decline in the number of patients, right? Because you're curing the patients. So getting that capacity right, it really is like catching a knife. If you don't have enough, you fail because you haven't distributed to patients and the market moves on. If you have too much capacity, then the idle capacity burdens each product, and the costs go skyrocketing. So it's almost not even worth building the cost from the bottom up. It's a business model question that may even say, okay, if we've got 1,000 patients, maybe we outsource 500 of them. We know that's going to be the volume. It'll be on a per product basis, a little more expensive to do that, but at least we know we have that fixed, and the developer themselves takes on the variability for that next 500 or 200 or 100, depending upon what it is, or you flip that equation around. But it's really got to be done on a case-by-case basis.
Yeah. Okay. Anybody else on the panel want to comment on that?
Yeah, this is Dave D'Augusto. I agree completely with Rob about the case-by-case basis considerations and the metrics he's used to illustrate those. One thing I'd also like to add is that timing has a lot to do with it, because in order to build in-house capacity, identify and or train expertise is a significant factor when it comes to manufacturing for a small company who may have limited resources and be on a very tight timeline before they burn through those resources. And so in that case, a CDMO or CMO brings existing expertise to the table, existing quality systems and existing plant facilities can really shorten a timeline. And that's really critical for small biotechs, for example, to get that proof of concept to keep the company alive, and then go for it'd be much easier to get additional funding after proof of concept. So I think that's a major consideration for many people, is the timing related to farming things out. Now, having said that, tech transfer again can go quickly if it's a common procedure like culturing CAR T cells. It may take a bit longer if it's a more complicated process like generating differentiated cells from iPS or ES source. So those are also considerations in how much time do you really save in the tech transfer part. But I think those are things I've considered both in an academic setting and an industrial setting that have impacted the choice to outsource or do it in-house.
Great points. Thank you. I think we can move on to our next topic here, which is on deal making. So for several years now, we've seen therapeutics developers become highly sought after acquisition targets, and 2019 has been no different. Roche announced a deal to buy Spark Therapeutics for nearly $5 billion. Biogen is acquiring Nightstar, and Vertex announced a pair of deals, including Exonics and Semma Therapeutics, and we're lucky to have David's perspective today. But in 2019 as well, there's been a wave of consolidation and deal making around cell and gene therapy manufacturing companies. So not only are we seeing therapeutics developers become targets, but manufacturing companies are now highly sought after targets as well in pretty substantial deals. For example, Thermo Fisher Scientific buying Brammer Bio for $1.7 billion and bringing in viral vector capabilities. Danaher acquired GE Life Sciences biopharma business, which included a cell therapy, cell processing platform, and that was a $21 billion deal. So I'd like to maybe start off with Bob and get your perspective. We're lucky to also have you on this panel as part of Hitachi, as Hitachi was involved in one of these key deals this year, buying Abscess, and also as part of PCT when that itself was fully acquired by Hitachi in 2017. So I'd love to get your thoughts on what you think are the factors that are driving this wave of deal making, and do you expect to see this pace of deal activity for manufacturers going forward?
Yeah, so I can only speak broadly for the industry as a whole, but more specifically what's been driving, as you mentioned, growth by consolidation and M&A at Hitachi Chemical for its cell and gene therapy manufacturing. We have a strong belief here that the therapeutic developers increasingly need access to multiple regions of the world, and the benefit of a harmonized network that offers them this access is a more efficient and consistent manner than working with separate providers or a loosely affiliated network. And so that's been driving a lot of the M&A activity for us. But in addition to that, that we call tier one of our strategy. Tier two of our strategy is to build a platform that changes that paradigm. It changes that quality, cost, scalability, and sustainability formula and brings some robustness to this process so that we can actually sustain manufacturing on these products through the commercial life of the product and through larger indications, which is where this is currently moving. And to do that, we have certain in-house capacity to develop applications. We have a lot of insight into that because we're doing manufacturing development, which includes process development and analytical development, and we're doing a lot of manufacturing, right, in different regions of the world. So we're really seeing where the pain points are, so we can put together that, biologics equivalent to a platform. We're in a very unique position to do that, but we're not a productizing type of a company. So we've got to go out there and not only add services, that's another place, because point to point, what's the value proposition for companies such as ours? And we're not currently doing any viral vector manufacturing. It sure seems like something we should be thinking about. There's non-viral gene editing technologies, there's analytical capabilities, all of these that we have built to various degrees internally or not, and we're looking outside to see what's the next place that can go. So I expect to see, to answer the end of your question, I expect to see this consolidation trend really increasing, and the bigger players, such as ourselves and the Lonzas of the world, and the big players are going around snapping up other parts to make more complete offerings to keep the geographic expansion and development of these products going.
Mm-hmm. Yep. Very interesting. Thanks for it. It's interesting to get your perspective. Michelle, I was wondering if you also had any comments, because earlier this year the majority of Aldevron was acquired by investment firm EQT. Do you have any comments in general on this wave of deal making that we've seen around manufacturing companies in cell and gene therapies?
Yeah, thanks, Amanda. So I think this really ties in with the previous question as well, in that you're really having more and more interest in how do companies, how do entities control their own destiny, and I think part of that, of course, is to bolt on a CDMO such as ourselves or other viral vector manufacturers. So I do think that is something that will continue, much to Bob's point. As for our particular situation, we're very proud of the relationship that has been now established with EQT in that with a private equity firm that has participated, we are now further enabled to service all. So we're not necessarily aligned exactly with one singular entity or viral vector manufacturer, that we can continue to really truly serve all. And that's the best place that we feel that we can be as a CDMO in our place.
Great. Thanks, Michelle. And David, did you have any comments? I know the Sematherapeutics deal is more of a therapeutics driven, but did you have any other any comments in general on the pace of deal making activity?
Well, I think you're going to see larger pharma starting to eat up some of these smaller companies, and it varies. Like Semma, we were lucky enough to be purchased preclinically, but as proof of concept comes out, I think big pharma is hungry for newer types of therapeutics, and I think the small molecule, low-hanging fruit's been picked. And so given the pace and potential for cell therapies, I think you'll continue to see sort of absorbance of smaller entities by larger ones. I have to say, for me, it's a really good thing, because I now have the resources I need to move to the next stage, which is clinical investigation. So it's much easier to do with a big pharma partner than to do on my own as a small biotech. So I think it's a good thing for the industry, and I think you'll continue to see both acquisition of technologies, including acquisition of capacity to manufacture those technologies, as critical deals that are going to be happening over the next 10, 15 years.
Okay, great. Yeah, thanks everyone. Really interesting discussion on that and deal making. So we'll move on to our next topic, which is around innovation. We've talked a little bit about this earlier. So Peter Marks, the director at FDA CBER, announced at the DIA meeting over the summer the formation of the CBER Advanced Technologies team with a goal of promoting the adoption of innovative manufacturing technologies for cell and gene therapy products through early interactions with the FDA. And innovative manufacturing has been a topic of interest for quite a while. So I'd like to get input from the entire panel on what you think are the barriers to innovating in manufacturing in cell and gene therapies, and also what are some of the new and innovative ways that companies are investigating and considering for manufacturing cell and gene therapies for the future. So I'll turn it over to the entire panel for your thoughts on that.
Maybe this is Mike from ARM. Maybe I'll just take the first crack at that. And one thing we're hearing a lot about is companies are looking to move CAR T or TCR type therapies into the solid tumor area. Are the challenges required there, because these really are in many ways personalized or customized therapies. And that means, for example, in the case of a particular tumor, or even for a particular patient, you may need to screen for a large panel of neoantigens, for example, to find out what the appropriate target in that tumor really is. And that could be different among different patients, and certainly is different among different tumor types. So I think one thing that is needed are more high throughput methods for dealing with these customized therapies.
Mm-hmm. Thanks, Mike. Anybody else have any thoughts on innovation in cell and gene therapy? I think it was Bob at the beginning talked about moving to automated processes, moving away from open manual processes. Is there any innovation going on there that that in your work experience?
Yeah, there's a number of different approaches that are going on, and it's distinguished, again, something David talked about before, between sort of off-the-shelf therapies and patient-specific cell therapies. There's slightly different approaches there. Specifically speaking, one of the bigger challenges is the patient-specific cell therapy, where the labor costs, the mistakes that are made, those sort of things hurt the quality because they're all manual processes with paperwork and, paper batch records and the like today, which is driving some of the manufacturing costs, which cuts into the profitability of the products and clearly makes scaling them difficult. And then the sustainability issues, again, talking about the reagents, the single source providers for reagents and equipment. So we have to address all of those, and it's more than automation. It really, we like to think of it more as innovation around, how do we address that problem, particularly when the end products are difficult to fully characterize and test. So the changes you make in the manufacturing process have the potential to change the biologic activity. And we don't always have a perfect handle on what are those markers that tell us that. So our approach has been, I think a large part of the industry approach has been to look at this on a unit operation by unit operation basis, which are easier to understand. If you can't sort of, draw the whole elephant, just take little pieces of that and then look at each one of those and apply that formula of quality, cost, scalability, and sustainability to each piece and evaluate the risks of what we talk about are the 5 M's, the man, the human component, the milieu, the environment, the materials, the methods, and the machines that are used, and apply the lens to each one of those and look at where the risks are. And I think ideally, the manufacturing platforms of today would be able to scale on a unit operation basis to accommodate all comers. So instead of having for each recipe, instead of having different machines, have machines that have wide capability, and then ways in which you evaluate the inputs and the outputs of that, and have, there's a lot of data analytics and the like, artificial intelligence going on here, with an underlying business process that ties the whole thing together. So it's a little more than innovation, and it might be a little bit early to lock a process, to put a process into its own instrument built on a bespoke basis for it, because the processes will change. They're not quite ready yet. And so the more flexibility you have until we get into the, you've done this 10,000 times now and you got it, now lock it up. I think, the platform's got to be more modular.
Yeah, this is Dave. I make a comment here because I think it's relevant to what Rob was saying, is that there's a market component to this, and for years before there was an approved cell therapy product, we were all borrowing from the blood banking industry and from the bioprocessing, say, on protein production industry for bioreactors, because no one was really committing to a cell therapy market. Now with the approval of, for example, a couple CAR T therapies and a gene therapy in the U.S., you're seeing that many vendors are willing to make the commitment because they see the market now. And so I think there's something that goes along with success in the field that will drive the willingness of manufacturers to create automated solutions or to create reagent sources for improving manufacturing. But in the absence of a projectable profit in doing so, I think we're going to move slowly in getting customized or advanced cell therapy specific automation into the field.
Mm-hmm. Great, yeah, great points. Thank you, Dave, Bob, and Mike for those thoughts. We can move on to our fifth topic here, which is on standards for new cell and gene therapies. So the FDA has urged manufacturers to rely more on standards, saying that it will ease the development and approval process, but it seems like developing standards and actually agreeing on standards in cell and gene therapy has been a slow process. And while the FDA has co-sponsored several workshops, including with the National Institute of Standards and Technology, the Standards Coordinating Body for Regenerative Medicine published a report earlier this year identifying a gap in certain areas such as cell counting, cell characterization, and raw materials, ultimately concluding that there is a need for more standards in cell and gene therapy. So maybe, Mike, I'll start with you, get your thoughts on ARM's perspective due to its work with the Standards Coordinating Body on this issue of standardization in cell and gene therapy.
Sure. So, there are a lot of great standards development organizations that exist. ISO, ASTM, USP, but and they're starting to work in this area, but there isn't a lot of experience in development of standards for cell and gene therapy. So that was the reason that ARM founded the SCB, the Standards Coordinating Body, which you mentioned back in 2017. SCB isn't a standards development organization per se, but they work to coordinate the efforts of other standards bodies, focusing specifically on standards for cell and gene therapy. And I certainly think we're in an early enough stage in this industry where standardization will benefit everyone. It's a case where a rising tide will lift all boats. So I would really, but these standards have to be right. I mean, the worst thing you can have is a standard that is not well developed and well thought out. So I would really encourage everyone who is listening in on this webinar to get involved with groups like the SCB and standards development. They're doing some great work on cell enumeration, as you said, and also on apheresis, which is a very important topic where standardization is needed. And I think this is just an area where we all need to work together.
Yeah. Thanks. It's interesting to hear, ARM's perspective on that and the work with the standards coordinating body. Michelle, Bob, and Dave, do you have any thoughts on standardization and what you think are the biggest gaps, and how do you think that these gaps in deciding on standards in cell and gene therapy should get filled?
This is Dave. I have a particular issue around genome editing, and it's interesting that the FDA has currently been asking for whole genome sequencing as a standard for determining off-target activity. And there's two problems I have with that. I think it would be helpful to have standards for this. One is there are a large number of possible off-target activities, but considering where off-target cutting happens in intronic and intergenic regions, it's not clear that these are necessarily actionable things when you find them. No one knows the consequence, so really having that as a standard for assessment of a product is hard to interpret in terms of the safety. And then the other thing is with current generations of, for example, CRISPR genome editing, you tend to get more changes in the genome from culturing the cells than actually manipulating with CRISPR. So what's hard, and I think would be very helpful for a field that's really starting to employ the use of genome editing technologies, to have a standard by which you can evaluate the safety of the genetic modifications that is very well defined and includes actionable results. For example, oncopanels, where a change in a particular gene will lead to, or is highly likely to lead to, a transformation event. So that's something I'm just commenting on. I think the field would benefit greatly from establishing some standards for safety assessment of these types of products.
Mm-hmm. Yeah, as we've had so many new company formation startups in this field of genome editing, that seems like a critical issue to address. So thank you for identifying that. Before we move on, just want to check to see, Bob or Michelle, if you had any comments on or insights on standardization.
Yeah, Amanda, this is Michelle. Sorry, Bob.
No, please, Michelle. Thanks.
All right, thank you. Just to comment from the perspective of what questions and things that we're getting from our client base here at Aldevron with regard to the plasmids themselves, and how they're viewed, what QC requirements, et cetera, are applied to them and how they're made. It's really been interesting because people are coming to us looking for almost a direction of sort, and it's difficult to provide that from our perspective simply because the material that we're providing is utilized in so many different downstream types of applications. Even considering within AAV delivery, we have clients who are delivering, of course, to the eye, of course, to via IV, intrathecal, et cetera. So it's almost a consideration within each group that has to be determined. So we are almost in that reverse position where we can't provide what that standard is. And so I think that's been an interesting perspective that we've been able to share. So I just wanted to provide that thought.
Thanks, Michelle. And Bob, did you have something?
So only to double up on what Michael said about joining groups that are working on this. ARM has launched the ACEL and A-Gene work streams to try to address these difficult issues that David and Michelle eloquently talked about. It's not an easy challenge here, but they figured it out in the biologics industry through the AMAP work stream. Came up with standards. Standards become increasingly more relevant and important as the fields mature, and the fact that FDA as a partner is encouraging us to come up with these really helps focus the mind. So it just encourages people to join these work streams.
Great. Yeah. Thank you, Bob, for mentioning that, and thank you for all of your inputs on this topic. Moving on to the last couple of topics we have are on specific challenges in manufacturing and how to overcome them. So starting with cell therapies, we know logistics in cell therapy manufacturing comes with several hurdles, such as the fact that you're dealing with cell availability issues, shorter shelf life, having a quality starting material that meets specifications, and many others. So hoping to get, in particular, David's perspective from Semma Therapeutics and Bob's perspective from Hitachi on what you think are the key challenges, as well as the solutions to some of the logistical problems that you've come across in cell therapy manufacturing.
Sure, and this is Dave. I actually prepared a list because I have a long list. But some of the challenges, we are encountering are things like supply chain management. How do we go up to, we go from our, tens of liters scale to hundreds of liter scale, presents a supply chain management challenge. Again, as you mentioned, the short shelf life of products as they're manufactured for a patient are delivered fresh and have to be shipped and infused in a very short amount of time. That presents logistical challenges. I think other ones that people don't think about is kind of more frequently is a logistic is the availability of experienced cell manufacturing talent. So as you scale up and you want to start making a lot more products, in many cases you're going to need more people, automation notwithstanding. And I think there is sort of a gap here in terms of the availability of cell manufacturing talent, and also the availability of qualified reagents. In many cases there are fully qualified reagents, but I think many of us experience that in a lot of cases we're still forced to use RUO reagents or research use only reagents, and that becomes a logistical challenge to go from a research reagent to both having a manufacturer making a GMP version, and then in many cases the performance of that reagent changes when it becomes a GMP qualified reagent. So those are some of the top things I've seen as challenges in the kind of manufacturing area.
Yeah. Thanks for that. Bob, did you have any comments on this?
I did, and I think, it was actually David's comment number 2, it was workforce development. Really it's about technical staff, manufacturing development, personnel, engineering teams, all of these people, and the infrastructure is developing. The solutions to these types of problems are forming directly around the unique challenges that are presented by this industry. And from community colleges around the country now to college curricula themselves and to graduate programs, there are biomedical engineering all the way down through just technical how to move around a clean room programs that are starting. This is a major pinch point in the industry today. Getting people in an environment with a low unemployment anyway, in an exploding industry particularly where these pockets that are developed of companies that do similar types of work, you've got people coming in and leaving at alarming rates. There's a lot of training going on. This is a situation that's got to be straightened out, and I think it is. I think there's an awful lot of work going on now to find ways to train people before they're coming into the environment such as ours, not just at a contract manufacturing level, but just into the industry, and how to retain those staff and how to keep them moving. There are all these very young, bright people who have opportunity left and right. So I think that's a major challenge, that we really just continue to keep our eyes on it. It'll hold us back.
Yeah. And it's Mike, if I could just jump in and echo that. I hear about workforce development issues almost every day now when I'm speaking to our members, and I agree it's absolutely a challenge, and companies are for the most part today having to deal with it themselves and investing a lot. It can take months to train an operator, and then when you lose them or they go to another company, you've lost that investment. So it's a big issue.
Yeah,
Mike, isn't ARM Foundation's focus, or one of their pillars, is focusing on infrastructure and how to tackle that workforce development piece and build that up? And
for sure,
it's a great initiative.
The ARM Foundation is looking at education in general. Not only education of the public and policymakers, but evaluating whether there's a role for the foundation or ARM to be played here in workforce development. I will point out one thing. I think it was Dave earlier that mentioned aging and ACEL, and thank you for bringing those up. We look at those best practice guides, which are built off the earlier AMAB example, as educational pieces, among other things. So as you have new people coming into the industry who, might understand quality by design, but they don't understand cell and gene therapy, or on the other side, people who have a lot of great cell therapy experience, but maybe they've never scaled a process up, they'll be able to use these guides, or companies will be able to use them to help educate their workforce.
Great.
There's something I wanted to add with this. Sorry, I was just going to mention, alongside of the manufacturing, I think it's important to just mention that the challenge with the analytics at a company, and that entities need to be thinking about their assays all the while, because they are very specific, and the time to do qualification, the time to do validation is not insignificant.
Great. Thank you, everyone. I appreciate your insights in bringing up those important issues, particularly with the workforce development. We do have one more topic. We only have a few minutes left, so maybe actually what I'll do is I'll just jump to the second part here, looking to the future. And Michelle, I'd love to get your final inputs on this, talking about, especially from your experience with Aldevron, discussing, if you could discuss next generation materials and non-viral delivery methods where you see potential in cell and gene therapies.
Yeah, so the challenge typically has been how to just deliver, non-viral DNA, and gene therapy has been around for quite some time, but I think that the delivery mechanisms have been slowly improving all the while, and I do believe that we'll see a pretty nice wave of successes coming through in the near term here, because of the innovation that's been taking place. Things such as electroporation, et cetera, have been slowly improving all the while in the background of advent of AAV and Lenti. So, my prediction is that this is going to be an even bigger impact in the coming months and years.
Mm-hmm. Okay, great. Thanks, Michelle, for that. So we are just out of time, I think, so I'll call the webinar to a close. I'd like to thank Mike Lemke, Bob Preddy, Dave DeGusto, and Michelle Berg for participating today and leading this important and interesting discussion on cell and gene therapy manufacturing. As a reminder, following the webinar, all registrants will receive an audio recording of the webinar along with a copy of the presentation. Thank you all again for joining today, and I hope you enjoy the rest of your day.
Thanks, everyone.
Thank you, everyone.
Thank you.